Hello everyone. My name is Rubesh Raja, and it's a pleasure to be here today to speak with you. My presentation, Model-Informed Drug Development, Deciphering Complexity for Better Decisions, will delve into how advanced modeling approaches can transform our understanding and decision-making in drug development. My modeling philosophy is built on 3 pillars: addressing R&D complexity. We embrace complexity by combining rich data with sophisticated models to uncover deeper insights. It's not just about data, but what we build with the data. Iterative dialogue. This refers to a continuous feedback loop between data and models. Our models aren't static. They evolve as new data emerges, constantly refining our understanding. Achieving actionable goals. The ultimate objective is to provide mechanistic insights that lead to actionable decisions. We aim to move beyond mere observations to truly understand why things happen, enabling more informed and impactful choices in drug development. To achieve these goals, I utilize a robust framework for Quantitative Systems Pharmacology, QSP, which integrates several advanced methodologies. Cybernetic Inspired Method, CIM. This approach focuses on goal-oriented regulation within biological systems, understanding how they adapt and respond to maintain function. Multiscale Modeling. We bridge different biological scales, from molecular and cellular interactions right up to systemic effects, to capture a holistic picture. Information Theory. By applying principles from information theory, we infer causal networks, identifying true cause and effect relationships rather than just correlations. Hybrid Stochastic Deterministic Modeling. This allows us to capture both the random, probabilistic events at finer scales and the predictable, macroscopic dynamics. Robust data integration and validation. Ensuring our models are grounded in high-quality data and rigorously validated against experimental observations is paramount. In this presentation, I will share examples of applying these frameworks across scales and data types. I am also ready to learn new methods or work with existing models, because the outcome, not the process, is what truly matters. My expertise in QSP spans the entire biopharma, R&D pipeline. In drug discovery, we can identify novel targets and understand their mechanisms of action. For preclinical stages, the focus shifts to optimizing molecules. In clinical pharmacology, we aid in determining optimal dose and regimen. And during clinical development, we assist with trial design and patient stratification for more effective outcomes. This presentation is divided into 2 parts. Part 1 will explore uncovering disease mechanisms, focusing on drug discovery and preclinical applications across therapeutic areas like oncology, immunology, diabetes, and novel modalities. Part 2 will then delve into optimizing therapies, covering clinical pharmacology and development. Let's begin with Part 1, where we leverage modeling to uncover the intricate mechanisms of diseases, which is crucial for early-stage drug discovery and preclinical development. Our first case study is in oncology, specifically focusing on cell cycle regulation. The core problem here is the inherent complexity of cell cycle networks and the challenge of establishing clear causality, understanding which events directly drive others. We leveraged high-resolution RNA sequencing time series data to capture dynamic changes. The goal was to reverse engineer the cell cycle to identify critical regulatory points. The application is directly relevant to drug discovery, helping us pinpoint the right target for therapeutic intervention. The diagram illustrates the different phases of the cell cycle: GON, S, G2, and M, and the genetic events that characterize each, which our models aim to decipher. To address this, we employed the Cybernetic Inspired Method, CIM, framework, a multi-step approach for regulatory networks. It primarily uses transcriptomics time series data to understand gene expression changes over time. The framework is designed to identify key regulatory molecules and integrate diverse biological information, including pathways, genes, interactions, and modifications. Crucially, it captures temporal changes such as chromatin remodeling, which are vital for understanding dynamic biological processes. The workflow at the bottom of the slide visually outlines these steps, from unbiased gene selection to constructing a simplified network. The first step in our CIM analysis involved significant data reduction. From the initial high-resolution data, we identified 4,248 differentially expressed genes. We then narrowed our focus to a more manageable set of 86 canonical cell cycle genes and transcription factor genes. This focused approach allows us to concentrate on the most relevant players in cell cycle regulation, as highlighted in the network diagrams. Moving beyond simple correlations, our CIM framework uses information-theoretic methods to link genes with cause-effect relationships. This supports biologically realistic objectives by understanding regulatory logic, not just Q expression. Specifically, we applied time-delayed mutual information, TDMI. TDMI is critical because it helps capture hidden gene relationships beyond raw correlations, providing a more accurate picture of gene regulation. The diagram illustrates how these causal links form the basis of our network. The next stage of the CIM framework involves using a cybernetic ODE, ordinary differential equation, model to refine these transcriptomics networks. We introduced a variable G as a lumped regulator of gene expression, with activation or repression determined by TDMI. Our objective was to maximize the production of molecules essential for cell cycle function. We applied a generalized weighted maximization and incorporated change point detection to identify abrupt transitions in the time series data. A significant finding from this cybernetic fitting was the revelation of 3 distinct S-phase stages, which had not been previously identified, providing a deeper mechanistic understanding. This slide visually demonstrates the power of the CIM framework to capture complex biological dynamics. The multiple plots show the time series data, dots, alongside the stage-specific cybernetic model fits, lines. You can see how accurately our model tracks the changes in RNA fold expression for various genes across different time points, reinforcing its predictive capability. Here we see the results of our network refinement, which made the model both mechanistic and dynamic. On the left, we have the initial network with 86 nodes and 731 interactions. Through our cybernetic modeling, we refined this to a network, right, with the same 86 nodes, but significantly reduced to 153 interactions. The Kijai term defines these gene-gene interactions, with arrows indicating activation and dashes indicating repression. The line thickness is proportional to the strength of this interaction. Critically, validation showed that 90% of these interactions matched known protein interactions, demonstrating the biological relevance and accuracy of our refined network. Having achieved a mechanistic and dynamically validated network, our next step was to perform further network simplification. This aims to distill the core regulatory elements without losing critical information. For this simplification, we employed graph theory metrics, maximal clique centrality, MCC, and maximal RNA expression rate, MRER. 1,200 measures note importance by the size of the largest clique it belongs to. Cliques are sets of mutually connected nodes. The interpretation is that larger cliques represent more central and important nodes within the network. We successfully reduced the network from 86 nodes and 153 interactions to 66 nodes and 126 interactions, retaining only the most critical regulatory components while maintaining a 12 score of 7 or higher in MRER greater than 1, indicating strong functional relevance. The impact of this cell cycle regulation study is substantial. We successfully distilled a complex network into 126 critical links from over 700, making it far more interpretable. This led to discoveries of novel regulatory interactions, such as between C-cone and BAT3. For validation, our model accurately predicted future cell cycles, a strong self-validation. The ultimate application is directly in drug discovery, enabling the identification of the right target with high confidence. I led all aspects, from conceptualization to model implementation and manuscript drafting. Next, let's explore an application in anti-infectives, focusing on bacterial signaling thresholds. The central question we addressed was, how do bacteria avoid overreacting to weak signals within their two-component systems, TCS? We encountered an intriguing data puzzle. The histidine kinase, HKAP, was observed to bind non-cognate response regulators, RS, more strongly than expected. This research has a direct application in drug discovery for novel modalities, particularly in identifying new ways to disrupt bacterial communication. The diagram illustrates a typical bacterial two-component signaling system. Our approach involved developing a mathematical model of TCS signaling. This model incorporated positive autoregulation and the role of non-cognate RS, using an ODE system to describe phosphorylation and gene expression dynamics. We analyzed various aspects of the signal, including maximum output, Omax, and total output, OTotal, in response to different stimulus inputs. Model fitting was achieved using global optimization in MATLAB, with AIC for model selection. Validation was rigorous, involving both in vitro phosphotransfer assays and in vivo tunable gene expression using M-Bovis BCG. The major impact was the identification of a sequestration sponge motif, a novel mechanism explaining how bacteria finely tune their response to environmental cues, preventing overreaction to weak signals. Our third case study on immunology, specifically in the area of metabolic regulation. The central problem is maintaining macrophage inflammation balance, a critical aspect of immune health relevant to various diseases, including diabetes. A key challenge we identified was enzyme A competition between arachidonic acid, AAA, and eicosapentaenoic acid, EPA, pathways, which produce pro- and anti-inflammatory mediators. We utilized metabolite measurements of prostaglandins, PGS, in macrophages as our primary data. The diagram illustrates the intricate metabolic pathways involved. For metabolic regulation, we again employed a cybernetic model, a complex system comprising 13 odes and 41 parameters. Our objective was to maximize combined pro- and anti-inflammatory responses, aiming for balance. The model used cybernetic control variables, UV, to represent implicit regulation, and incorporated nonlinear kinetics to accurately describe metabolite rate balance. A significant outcome was the prediction of unmeasured enzyme profiles, such as COX. Parameter estimation used a hybrid optimization approach, pattern search plus fmincon, and validation included ftest. Leave one out cross-validation and testing against unseen scenarios, ensuring model robustness. The graphs on the right show excellent agreement between model predictions and experimental data. The impact of our metabolic regulation study was profound. The model successfully captured complex nonlinear trends that traditional Michaelis-Menten kinetics failed to explain. A key finding was the identification of a dynamical switch between pro- and anti-inflammatory states. This work directly informs dietary recommendations, particularly concerning the optimal AAEPA, which is crucial for modulating inflammatory responses. The application extends to a deeper disease understanding in areas like immunology and diabetes. Moving to novel modalities, we explored the emerging area of microbiome and gut health. This field is rapidly evolving, focusing on the complex gut microbiome dynamics. The main challenge lies in understanding the complex microbial interactions and how they are influenced by diet. Here, too, cybernetic models prove to be a highly effective methodology for tackling this intricate biological system. The cybernetic model applied here for microbiome studies has a powerful core. It focuses on optimal growth decisions by microbial species, governed by control variables. It's a generalized framework capable of modeling multi-species, multi-substrate growth with extracellular hydrolysis. This allows for strategic modeling, distinguishing between competitive and cooperative behaviors within a microbial community. We also utilized an advanced tool, PAMA GCMs, for precisely tracking substrate linkages, which is crucial for validating the model's predictions. The equations and substrate matrix illustrate the mathematical foundation of this generalized model. To support our modeling, we conducted synthetic community experiments. These involve diverse microbial cultures and various carbon sources like FOS, AX, and EPS, generating extensive data. Our modeling approach used the generalized cybernetic model for multi-species, multi-substrate growth. The key goal was to unravel the competitive versus cooperative interactions that dictate community behavior. The ultimate impact of this work is in enabling personalized nutrition strategies by understanding how an individual's microbiome responds to dietary interventions. Our final Part 1 case study addresses the critical issue of antibiotic resistance. AMR is not a distant threat, but a daily reality. Efflux pumps are a key bacterial survival mechanism, actively expelling antibiotics and driving multi-drug resistance. Our research focus was on understanding the regulation of these efflux pumps under antibiotic pressure, aiming to identify vulnerabilities. In a CO-authored study, we investigated how these pumps are regulated under antibiotic pressure, work that directly supports drug discovery and novel therapeutic strategies. Building on the AMR threat, our study specifically aimed to quantify efflux pump-mediated antimicrobial resistance. Efflux pumps are not only crucial for bacterial survival, but also represent a tunable resistance mechanism. Our approach combined mathematical modeling with experiments to achieve our goal. Quantifying the maximum resistance, mic-fold change, resulting from efflux pump-up regulation. The diagrams illustrate the mechanism of drug efflux in the absence and presence of a drug. When a drug binds to repressor proteins, like LFRR in Mycobacterium smegmatis, it lifts repression and triggers efflux, pump expression. The pumps, like LARA, then actively expel the drug, creating a rapid, tunable, and reversible form of phenotypic resistance. But with prolonged exposure, bacteria often acquire mutations that permanently disable repression, leading to constitutive efflux pump overexpression, something frequently observed in clinical isolates. In our study, we set out to quantify just how much resistance this mechanism can generate. Using M. smegmatis and ethidium bromide as a model system, we combined mathematical modeling with experimental validation to measure both the transient phenotypic induction and the permanent genotypic disruption of efflux regulation. This gives us a clearer picture of the limits of efflux-mediated resistance and a foundation for targeting it in drug discovery. Our integrated model captured coupled drug transport, efflux regulation, and MIC prediction. It was rigorously validated by accurately capturing ethidium bromide, EDBR, dynamics, and mics. Key findings revealed a phenotypic induction that nearly doubled the minimum inhibitory concentration, MIC, with maximum resistance reaching 14 UGA/mL, a twofold increase over wild type. The model also showed that resistance is highly sensitive to influx-efflux rates and protein synthesis. This provides crucial intervention potential. Targeting efflux pump inhibitors or membrane permeabilizers could be effective strategies against resistance. To quickly recap Part 1, we've covered 5 diverse applications of our model-informed approach in uncovering disease mechanisms. From oncology and cell cycle regulation, where we reverse-engineered complex networks, to anti-infectives, understanding bacterial signaling thresholds. In immunology, diabetes, modeling metabolic regulation for inflammatory balance, exploring novel modalities in microbiome and gut health, dissecting competitive and cooperative interactions. And finally, in antibiotic resistance, quantifying efflux pump mechanisms. Each case demonstrates the power of our methodologies to generate deep, actionable insights in drug discovery and preclinical stages. Now, let's transition to part 2, focusing on how our approaches are used in clinical pharmacology and clinical development to optimize therapies. Our first case study in part 2 focuses on hepatitis C virus, HCV, and the optimization of direct-acting antiviral DAA regimens. We identified a significant clinical paradox. Patients with prior interferon, IFN, treatment failure often exhibited lower DAA cure rates. The problem was to explain this surprising correlation between prior IFN response and DAA efficacy. We gathered extensive data from clinical studies on cure rates, viral load, and in vitro experiments. The application here is directly for clinical pharmacology, determining the right dose, and clinical development, designing optimal trials. The charts on the right illustrate these complex response patterns to various DAA regimens. To explain this paradox, we developed a multiscale model framework for HCV, starting at the cellular level. The cellular component focused on the HCV-FN interaction network, particularly a double negative feedback loop involving PKR. This mechanism led to a key result: bistability, meaning cells could exist in either a refractory or a responsive state to IFN. The model used ODEs for viral RNA and ISG protein dynamics. Validation was achieved through in vitro time of addition experiments, confirming the model's ability to capture cellular responses accurately. The diagram illustrates these cellular interactions and the bistable behavior. Building on the cellular insights, our HCV model extended to the individual and population levels. At the individual level, we used a modified viral kinetics model that incorporated 3 hepatocyte phenotypes derived from our cellular model. ODEs describe the dynamics of target cells, infected cells, and virions. At the population level, the model accounted for the distribution of individual IFN responsiveness, allowing us to predict population-level cure rates. Crucially, this multi-scale model achieved quantitative validation by matching data from over 50 clinical trials, demonstrating its robust predictive power across different scales. The HCV multi-scale model yielded significant impact and actionable insights for personalized dosing. It revealed that a strong IFN response limits replication space, which in turn constrains the emergence of drug-resistant mutants, RAVs. A key actionable insight was the ability to recommend personalized DAA treatment durations. For example, our model suggested that 20% of patients might achieve cure in 8 weeks, while 50% might require 10 weeks, compared to the standard 12-week regimen. This has direct application in clinical pharmacology for determining the right dose and in clinical development for designing optimal trials, potentially reducing treatment duration for many patients. Continuing with HCV, this case study addresses the critical problem of DAA failure due to hidden drug-resistant variants, RAVs. The challenge is that these RAVs often exist below the detection limit, N0.1%, in the patient population, making them difficult to identify clinically. We focused on understanding the HCV diversity and the spectrum of mutants. The application is vital for drug discovery and clinical development to help prevent resistance by anticipating mutational pathways. SHIV mutates before integration into host DNA, so each infected cell produces only one type of viral progeny. In contrast, HCV replicates in the cytoplasm, where errors accumulate continuously, allowing a spectrum of mutants to emerge within a single cell. To tackle the hidden RAVs, we developed a multiscale semi-stochastic hybrid methodology. The stochastic intracellular component utilized the Gillespie algorithm to model codon-level evolution, all nucleotide variants, estimating relative infectivity and specific release rates. This allows us to track rare events. The deterministic extracellular component employed a multi-strain ODE model in MATLAB, taking inputs from the intracellular model to track viral population dynamics. The key benefit of this hybrid approach is achieving high accuracy without prohibitive computational cost, making it practical for exploring a vast number of mutational possibilities. The diagrams illustrate how intracellular and extracellular processes are integrated. The impact of our HCV mutant model is significant. It allowed us to generate comprehensive mutational pathway maps, showing how resistance emerges. A key insight was the discovery of genotype-specific RAV prevalence. For instance, the R155K variant was found to be approximately 100 times lower in genotype 1B compared to 1A. These applications are crucial for designing optimal DAA combination therapies, informing clinical testing for early detection of resistance, and developing future vaccine strategies. Ultimately, this work aims to guide drug discovery and clinical development efforts to prevent resistance from undermining treatment success. As lead author, I drove the conceptualization, methods, analysis, and visualization that made this possible. Next, we move to a fascinating area, SHIV and broadly neutralizing antibodies, BNABs, for lasting control. The breakthrough here is the observation that short-term BNAB treatment can lead to lasting control of SHIV infection. This presented a scientific puzzle. How can a transient treatment result in a permanent outcome? Our data came from in vivo macaque studies, including viral load and immune cell data. The application of this research is in understanding the mechanism of action, MOA, and developing optimal strategies in immunology and novel modalities. The diagram illustrates the SHIV infection cycle and how BNABs intervene to shift towards viremic control. To unravel the SHIV puzzle, we employed a dynamic systems model based on ODEs. The model incorporated various components: infectious, non-infectious, and latent virus, target cells, activated and exhausted effector cells, and BNABs. A crucial aspect was modeling T-cell exhaustion using a Hill coefficient, NaNA4, and accounting for the pleiotropic effects of BNABs. Our model-building strategy involved gradual complexity addition, ensuring each layer was robust. Parameter estimation was performed using population-based nonlinear mixed effects, NLME, modeling, monolix, and global optimization, MATLAB. Rigorous validation included quantitative fitting to in vivo data and robustness analyses, confirming the model's accuracy. The SHIV model provided powerful insights. A key insight was that the system exhibits B-stability, which means it can switch between progressive disease and viremic control states. The mechanism for BNABs was identified as a dynamical switch by enhancing viral clearance and immune priming. This also highlighted an advantage over traditional antiretroviral therapy, RT. The immune priming component offered by BNABs could lead to lasting control even after treatment cessation. The application is crucial for understanding MOA, developing optimal strategies in immunology, and exploring novel modalities for HIV treatment. The graphs illustrate the bistable behavior and the sustained control achieved with BNAB treatment compared to ART. Our final case study focuses on sickle cell disease, SCD, and the challenge of personalized adherence to hydroxyurea, HU therapy. The problem with HU therapy is variable patient response coupled with high rates of non-adherence. A major challenge is distinguishing whether a patient's suboptimal response is due to underlying biology or simply non-adherence, especially since pharmacy records are often imperfect. We utilized longitudinal clinical biomarkers from 85 children in the HUSTLE trial. The application is in clinical pharmacology for achieving the right doses and in clinical development for optimal trial design, ensuring patients receive the full benefit of therapy. To address the SCD challenge, we developed an integrated pharmacokinetic pharmacodynamic (PK/PD) model. The PK component linked WHO dose to plasma concentration. The PD component modeled biomarker dynamics, including efficacy markers like HbF and 1105 using an adapted erythropoiesis model, and safety toxicity markers like ANC and RBC using a leukophoresis model. The model also calculated mean cell fetal hemoglobin (FM). Crucially, our dose calculation method utilized pharmacy refill records and explicitly accounted for missed or extra doses between clinic visits, providing a more realistic picture of adherence. Model fitting was performed using MATLAB functions like LS-Kanalan, fminCon, and pattern search. To further refine our understanding of known adherence, we introduced a novel probabilistic approach. Step 1. We initially fit the PK/PD model to pharmacy data to establish baseline parameters. Step 2. An iterative comparison was then performed between model predicted values, V model, and observed data for FEVA for 105. Step 3. The probability, P, was adjusted for missed doses when the model predicted 1/5 was greater than the observed data, indicating potential non-adherence. Step 4. A Bernoulli trial was used to generate a daily dosing profile based on this probability. Step 5. We then re-simulated the PK/PD model iteratively to minimize the sum of squared errors. SAE validation showed that this mid-V inferred dosing significantly improved the HbF fit, demonstrating the utility of our probabilistic method in accurately estimating real-world adherence. The flowchart visualizes this iterative process. The impact and clinical utility of our SCD model are substantial. It allows clinicians to distinguish between biological non-response and true known adherence, providing clarity for treatment decisions. A key actionable insight is the quantification of drug forgiveness, revealing that the pattern of missed doses truly matters. We found that drug holidays, consecutive misses, led to severe drops in therapeutic effect, whereas sporadic misses were less detrimental. This has direct application in clinical pharmacology for determining the right dose and in clinical development for designing optimal trials, enabling personalized counseling for adherence and improved patient outcomes. Finally, I want to briefly touch upon another area, multiscale immune modeling, particularly relevant for oncology and immunology. This work focuses on the immune response, specifically CD8 plus T cells, in the context of viral infections and cancer. The primary challenge is the immense complexity across multiple biological scales, from molecular interactions to cellular behavior and systemic responses. The ultimate application is to optimize immunotherapy in oncology, a field with immense potential. This slide also highlights a relevant advanced review on this topic. Our multiscale methodology for CD8 plus T cells is designed to explicitly decompose overall dynamics. It uses a hierarchical abstraction to effectively connect different biological scales. The model integrates intracellular signaling, single-cell behavior, and population-level immune responses into a coherent framework. The goal is to gain a deep understanding of the T-cell response and its role in viral clearance and control. The impact is significant. It allows us to predict the efficacy of immunotherapies and aid in vaccine designs, accelerating the development of more effective immune interventions. To quickly recap Part 2, we've explored 4 key areas where our modeling optimizes therapies. We began with HCV, optimizing DAA regimens through multiscale modeling for personalized dosing. Then we tackled HCV mutants, mapping resistance pathways with a hybrid approach to prevent treatment failure. We examined SHIV, studying BNABs for lasting control, and uncovering the mechanism of a dynamical switch. And finally, we addressed sickle cell disease, personalizing adherence to WHO therapy to improve patient outcomes. These examples highlight how our model-informed approach provides actionable insights throughout clinical development. In summary, my core competencies include strategic problem solving, the ability to dissect complex biological and clinical challenges into solvable modeling questions, advanced QSP modeling, expertise in methodologies like the Cybernetic Inspired Method, Multiscale, and hybrid modeling to provide comprehensive insights, robust data integration and causal inference, a commitment to grounding models in high-quality data and uncovering true cause and effect relationships, actionable R&D, strategies and tools, translating complex model outputs into clear, practical strategies and tools for drug development. A collaborative approach. My work thrives in interdisciplinary environments, fostering strong partnerships to achieve shared goals. Looking ahead, my vision for advancing model-informed drug development encompasses several exciting areas. Developing digital twins in biology and medicine, creating highly personalized in silico representations of patients to predict treatment responses. Decoding developmental processes, such as cell differentiation, using models to understand and potentially manipulate fundamental biological events. Championing ethical AI in healthcare by ensuring our models are transparent and interpretable, fostering trust and facilitating adoption. Ultimately, our goal is to continue driving a data-driven future for biopharma, R&D, accelerating the delivery of safe and effective medicines to patients.